Hello everybody. During this presentation, I will cover the rest of chapter 16, and more specifically the second half of the citric acid cycle. Also, I will talk about the regulation, and I will finish the presentation with the glyoxylate cycle. Last time I ended my presentation with this slide, and I asked the question, were the carbons of the acetyl-CoA that entered this cycle were released during the first half of the citric acid cycle. Now, if you look carefully, the carbons of the acetyl-CoA are highlighted in pink. If you follow the reactions of the citric acid cycle, you will notice that the first carbon dioxide that was released came not from the acetyl-CoA molecule that entered this particular cycle. The second carbon that was released later on was also not part of the acetyl-CoA molecule. Then where is it coming from? Where are these carbons coming from if they don't come from the acetyl-CoA? Well, obviously, they will come from the other member of the first reaction of the citric acid cycle, which is oxaloacetate. So basically, the acetyl-CoA that enters the cycle is not being degraded to carbon dioxide. Instead, oxaloacetate donates the first or the two carbon dioxides that are released during the citric acid cycle. So what happens with acetyl-CoA? Acetyl-CoA is being invested in a way or incorporated into the regeneration of oxaloacetate. So when will these carbons be released? It is actually unclear when would the carbon dioxides from this particular acetyl-CoA will be released. And why is that? Because after succinyl-CoA, the following molecule, the following compound, which we're going to talk about in a minute, is succinate. Succinate is a symmetric molecule. Fumarate is also a symmetric molecule. So how exactly the carbons donated from acetyl-CoA will be oriented, and which carbons will be in this position or this position is somewhat unclear, or it's hard to predict. We can tell for sure that one of the carbon dioxides from this acetyl-CoA will be released, but when will the next one be released? It will be hard to say. Perhaps in the third cycle, perhaps in the fourth cycle. It's hard to say. But for sure, one of these carbons will be released because if you follow which one was the first carbon, it was the carbon from, the carboxyl group that was bound to this carbon, and then which one's the next one is the one, I get confused. It was this carbon carboxyl group that was released, and then this carboxyl group was released. So it's either this or this carbon from oxaloacetate. Well, they both are released, but based on which of these two entities are contributed by this acetyl-CoA, we know that one will be released, but we cannot guarantee the other. I hope this made sense. The most important message, though, out of all of this is to realize that the carbon dioxide that is being released in the citric acid cycle is not directly from the acetyl-CoA molecule that enters the citric acid cycle, but these carbons come from oxaloacetate. Acetyl-CoA is being used to regenerate oxaloacetate, and the acetyl-CoA that enters this, let's say, this first cycle will donate or contribute to the carbon dioxide that is being released later in the following cycles, at least two more. All right. Then someone would say, then why do we say that the acetyl-CoA is broken down to carbon dioxide? Well, in essence, it is, because whether it's going to be broken down during the cycle that it enters or later on, it doesn't really matter, because oxaloacetate, if you look at it carefully, it is, well, in essence, looks like two acetyl-CoAs that are fused together. And that's what's important for us to realize at this point, that acetyl-CoA gets burned through this cycle, whether it's right away or a little later, it doesn't really matter. But the product of acetyl-CoA degradation is carbon dioxide. All right, so last time we ended with the generation of succinyl-CoA, and now we'll continue with the conversion of succinyl-CoA to succinate through succinyl-CoA synthetase. In this reaction, we're generating one molecule of GTP. And even though this reaction we're generating a high energy bond, we still have a reversible reaction. The energy of the energy of the delta G of the high energy bond the succinyl-CoA carries has a negative 36 kilojoules per mole, which is sufficient to generate a molecule of ATP, which, as you know, has a standard free energy change of 30.5. Now, the question I want to ask you, would you consider this reaction to be efficient? Now, what would help you realize that? Well, you know that the energy of the breaking of the succinyl-CoA bond, high energy bond, is 36 kilojoules per mole, and that the energy of formation of GTP or ATP is positive 30.5. The free energy that is being released is heat, in other words, that dissipates and we cannot recover it, is only 2.9. So majority of the energy released by the breaking down of succinyl-CoA has been invested in the ATP. This is, for all intents and purposes, this is a fairly efficient reaction. This process describes how this happens, and you can see that we have the help of inorganic phosphate that is involved in the phosphorylation of succinyl-CoA. And then the phosphate gets transferred to the enzyme, and then from the enzyme to GDP. Another question that you might ask is, well, what happened with the rest of the energy? We have 36, the ATP's production is 30.5, this is only 2.9, so there's a few kilojoules per mole that are unaccounted for. Well, I just want to tell you that this is not always perfect math, that we have energy sometimes invested in conformation changes within the enzyme, multiple steps, and sometimes energy that is involved in binding energy. It's hard to account for, to be precise. So even though the math is not perfect, you could, say that this is a fairly efficient reaction in terms of the energy that was harvested and that was reinvested into ATP. The following reaction is the conversion of succinate to fumarate by oxidation of succinate, catalyzed by succinate dehydrogenase. Again, a reversible reaction. It's not surprising that delta G is close to its zero in this case, so it's easy to move this reaction backward and forward. Something noteworthy, though, is what is the electron acceptor in this case. We haven't seen this so far. This is the first time we're talking about FAD. Well, actually, it's not the first time. Pyruvate dehydrogenase was the first time when we talked about FAD. But this is the second time, basically. So FAD accepts an electron pair and then is converted to FADH2. Something that we should note at this point, and you don't understand why this is the case, but we will understand this a little. I will explain this a little later. For each electron pair carried by FADH2, the cell can synthesize 1.5 ATP molecules, while for each pair carried by NADH, the cell can synthesize 2.5 molecules of ATP. So we get more ATP per electron pair carried by NADH2 than from FADH2, from NADH than from FADH2. Next reaction is the hydration of fumarate to malate catalyzed by the enzyme fumarase. The reaction is then followed by the oxidation of malate to oxaloacetate with the help of the enzyme known as malate dehydrogenase. The electron acceptor in this case is NAD plus converted to NADH. Now notice the delta G of this reaction, and then think about this statement that if allowed to reach equilibrium, the reaction will be far towards the substrate. Does that make sense? Of course it does. This reaction requires basically the energy from an ATP hydrolysis. Somebody has to donate this energy. This is not a favorable reaction. It has a high energy demand. That energy has to come from somewhere. Obviously, we're not told where the energy comes from. Probably from binding energy, probably somewhere from the enzyme. Whatever it is, that reaction, that energy has to be paid. Somebody has to provide that energy. Something has to provide that energy. And for that reason, this reaction is unfavorable and heavily in favor of malate. However, in the cell, the reaction is driven towards the product. Why is that? We've talked about a reaction like that in the past. And the explanation was that these reactions that normally are not driven towards the product, but in the cell, they do get extinguished and they do get completed towards the product. And that is facilitated because the product is somehow involved into a favorable follow-up reaction, which in this case is the citrate synthetase reaction, combining oxaloacetate with acetyl-CoA forming citrate, which is a very favorable irreversible reaction. So, if you remember, at the beginning of the cycle, I asked you, what do you think the concentration of oxaloacetate would be, high or low? And at that time, we speculated that, we know that it's low, but why was it low? Well, because there's no need, because oxaloacetate gets regenerated. Also, oxaloacetate gets used in a very favorable reaction, like the citrate synthetase reaction. Also, oxaloacetate can go back and be used for glucose synthesis through the gluconeogenesis pathway. But another reason why we don't have high oxaloacetate concentration is this. The reaction from malate to oxaloacetate is not favorable. This reaction is, the equilibrium is heavily in favor of malate. For that reason, the concentration of oxaloacetate, together with the other reasons, the concentration of oxaloacetate is low in the cell. Alright, I hope this made sense. It's a combination of factors. Unfavorable reaction that doesn't want to produce the product, followed by a favorable reaction that pulls away the product, together with the fact that oxaloacetate also can be used in other processes, causes for oxaloacetate, is the reason for oxaloacetate to have a low concentration in the cell. So here it is. We went through all the reactions of the citric acid cycle, and this is such a central pathway that you have to remember, or you have to know these reactions. Now again, similarly to the advice I've given you for glycolysis, the minimum that you could do, or at least that you could do, if you feel like I cannot remember all these names, at least know what happens in each step. Even if you cannot remember all the names, at least know what happens. Now, if you know that, I would say start adding the names. at least of the key reactions, know the reactions that produce carbon dioxide and electrons. What are the electron carriers? Do we make energy anywhere, such as in the production of GTP when succinyl-CoA is converted to succinate? And then start adding the names of the enzymes of these key reactions, the metabolites of these key reactions. I mean, whatever helps you, but this pathway is an important pathway, and you need to know it, and you need to know it really well. Because this is not going to be the end when we talked about it. We're going to keep coming back to this pathway. Okay, so someone might say, well, how do we know that all these things actually happen in the cell? How can we tell which carbon gets released, which carbon is added here or there? Like, for example, in here, how do we know that the acetyl-CoA actually is not degraded, that the carbon dioxide doesn't come from the immediate acetyl-CoA that was added, but comes from oxaloacetate? Well, in this research, the use of radioisotopes, specifically radioactive carbon or carbon-14, is the way to track the carbons or carbon processing. So in this particular case, if we use, that is described here, if we use carbon-14 radio-labeled acetate in the form of acetyl-CoA, and then we condense it with oxaloacetate, we're going to get labeled citrate. Now, the citrate is a symmetrical molecule. And if we isomerize citrate to isocitrate, it would lead to two equally represented forms of isocitrate, which when they're oxidized to alpha-ketoglutarate, again would lead to an equal amount of two alpha-ketoglutarate forms, one that is labeled at the gamma position and one that is labeled at the alpha position. So that's what we should expect. This is a symmetrical molecule. However, in the cell, only one form is produced, the one that is labeled at the gamma position. So how is this possible? I mean, anything you've learned in organic chemistry would say this is impossible. Why would only this product be observed in the cell and never this product, or the one that is labeled at the alpha position? Especially since citrate is a symmetric molecule. It doesn't have chiral centers. It is symmetrical, but it acts asymmetrically. Since, well, these type of molecules which are not symmetrical but act asymmetrically are known as prochiral molecules. So citrate is prochiral. That's not because of itself, but because what it binds it. So this is possible because the active site of the enzyme is asymmetric, meaning that it binds the substrate only at a specific orientation for 3 points, thus making only a specific bond available for a reaction. So to use the schematic on this slide, the hydroxyl group always has to bind at the X position, while the carboxyl group, this carboxyl group, has to bind always at the Y position, which positions only one and always the same one of the two identical carbon-carbon bonds at a position for a reaction. Now, in order for the other one to react, if you can visualize this, the hydroxyl and carboxyl groups must switch the XY positions, which is impossible. Basically, you have to have the hydroxyl bind Y and the carboxyl bind the X binding site. Now, this means that the orientation of the binding positions of the hydroxyl and carboxyl groups determine which one of the two, and always the same one of the two, identical bonds to be available for a reaction. This is yet another example how seemingly impossible reactions in biological systems are not only possible but the rule. This also demonstrates how 2D representations cannot help us see the truth as well as the 3D orientation. And even the 3D representations sometimes are not sufficient without the needed added complexity. These examples usually make me realize and humble me in a way and help me to realize and to be mindful and careful what I personally label and claim to be true, and to be especially careful when I call something false or untrue. Because using logic, this will be impossible, because we just thinking about it, this symmetrical molecule can bind either way, but there's something else that we are not seeing, that we couldn't know if we just look at these reactions like that, theoretically, from pure organic chemistry view. And again, it's not that organic chemistry cannot explain this. Of course it can, but if we don't know the requirement or the fact that the site of the enzyme is the one that acts asymmetrically and that makes this molecule be asymmetric, even though it is symmetric, we would not know, and we would not be able to explain why this is happening. These examples, I focus on these examples not because there's something, I want to use this example actually to hopefully basically convey a message that we as human beings should be careful when we label something to be wrong, because sometimes we don't really know the complexity of life or the complexity of truth. We should be careful when something is somewhat uncertain, or when we don't have 100% evidence for something, to claim that something is false or untrue. So be curious, but be careful when you claim what is true and what is not true. All right, so overall, what we can say is that each acyl-CoA, and again I'm putting this in quotation marks so that you know that it's not directly for the acyl-CoA that enters the cycle, but eventually each acyl-CoA gives 2 carbon dioxides. The cycle also generates 3 NADHs, 1 FADH2, and 1 ATP. So for each molecule of NADH, for each electron carried by NADH, we get 2.5 ATP molecules, and for each electron carried by FADH2, 1.5. Then what is the energy product from 1 molecule of glucose starting from glycolysis? You can see the reactions on this table. The grand total is 30-32. Someone might say, why not one number? And why either 30 or 32? Well, it's because of this reaction of glycolysis, where we produce the 2 molecules of NADH. We will understand this a little later. I'll tell you this a little later. It has to do with how these 2 electron pairs are delivered to the oxidative phosphorylation pathway, and that will determine the energy outcome of a single glucose molecule. All right. Now, when we, mathematically speaking, the maximum energy of oxidation of glucose is 2,840 kilojoules per mole. Now, based on the delta G of ATP, what is the amount of conserved energy? If we use the standard energy change of ATP, we will determine that about 34% of the energy of a glucose molecule is conserved as ATP. If we use the actual free energy change, we will determine that about 60% of the glucose molecule that has been oxidized and then invested in ATP has been preserved. Only about 60% of this energy is preserved as ATP. A good question would be, then where's the rest? Well, it's heat. The energy that was lost in a way, or that was not invested in ATP, was lost as heat. And part of it was also invested in binding energy, and most likely was invested in, dissipated as heat. So only about 40% of the glucose energy is used is converted into ATP. The rest of it is lost as heat. Okay, so with respect to the carbons, the cycle ends at succinyl-CoA. The carbons are released, which is the half point. Then why do we need the rest? And why such complexity? Now, if we only care about the carbons, well, we're done. Succinyl-CoA, we're done. All the carbons are released. Yeah, but if you think carefully, and if you look carefully, you will see that the half of the energy of the citric acid cycle is made in the second half. Also, acetyl-CoA is not the only entry point for metabolites. Alpha-ketoglutarate, oxaloacetate, they're generated by deamination of glutamate and aspartate. Well, transamination in this case because the amino group is transferred. But basically, if you take the amino group from glutamate and aspartate, you get alpha-ketoglutarate and oxaloacetate respectively. Now, metabolites of the citric acid cycle are also precursors for many biosynthetic processes. Because the citric acid cycle is catabolic and it's also involved in anabolic processes, we call this pathway to be an amphibolic pathway. In other words, applied to multiple types of metabolic pathways. I'm going to use this image to show you the complexity of the crosstalk between the citric acid cycle and other biosynthetic processes. And they are not all shown here, by the way. But I don't expect you to memorize this in any way. I just want you to appreciate why all reactions in the cycle are important, not just those that make energy or carbon dioxide. All right, so in terms of regulation, which steps do you think are regulated? Well, I'm listing them right here. Production of acetyl-CoA, production of citrate, isocitrate dehydrogenase, and alpha-ketoglutarate dehydrogenase. They seem to be all focused towards the beginning. Well, what is the significance of the first two? Well, the pyruvate dehydrogenase complex reaction, is a checkpoint. During this reaction, decides whether pyruvate will become acetyl-CoA, which then can be used for biosynthesis or for breaking down, or whether pyruvate will go back for gluconeogenesis. This is a very important reaction. The citrate reaction decides whether acetyl-CoA will be used for biosynthetic purposes, and we'll talk about those a little later, or whether it will go for fatty acid synthesis. In other words, this is the entry point of the citric acid cycle. the reaction has to have heavy regulation. This is a decision-making reaction. Once it enters, then it goes down for degradation. I mean, there are other checkpoints here with alpha-ketoglutarate and succinyl-CoA that are involved in other reactions, but for the most part, this reaction decides whether acetyl-CoA will enter or not. Now, which are the main activating factors? Well, in terms of activation, we can see that, from the schematic, basically, that calcium is activated. Someone would say, why calcium? To be completely honest, I'm not totally sure why. But another activator is ADP, and you can see that ADP here, AMP here, which is just also a product of ATP degradation. Also CoA, NAD+. Does this make sense? Of course it does. If you need energy, these reactions will enter, or these metabolites will enter. The citric acid cycle will be activated because we have to generate ATP. If we have high ADP and high AMP, that means we have low ATP, and we have to generate it. What are the main inhibitors? You can see all these red crosses, right? Inhibitors is high ATP and NADH, succinyl-CoA. These are all citrate. Well, citrate is, inhibitor of upstream reactions, but these are the inhibitors for the citric acid cycle. When we have high fatty acids, which are used for energy, that means that they will be degraded. You don't know that yet, but they will be degraded to acetyl-CoA. Having high acetyl-CoA will inhibit the pyruvate dehydrogenase reaction, telling glycolysis, stop. Use pyruvate to make glucose, because we can use now fat for energy instead. High ATP also allosterically activates a kinase, which phosphorylates the pyruvate dehydrogenase reaction and turns it off, telling the process, the glycolysis, stop. Don't degrade don't produce pyruvate, or use pyruvate for something else. Use pyruvate for glycogen production or something else, but there's no need to degrade pyruvate any further. Now, an interesting thought was also proposed when we talk about the citric acid cycle, or all metabolic pathways for that matter. And that is that these individual enzymes are not really by themselves floating into the cell, and they act for themselves. In other words, citrate is generated. At this point, citrate kind of floats around in the cytosol, and then the isocitrate dehydrogenase will catch it, and they'll convert it to, the aconitase will catch it and will convert it to isocitrate. And then isocitrate gets released, and then isocitrate dehydrogenase catches it at some point and converts it to alpha-ketoglutarate. This is a gigantic waste of space and time, if they are all just floating around without any order. It's actually being suggested and shown that these enzymes are all part of large enzyme complexes. And when one enzyme produces a product, that product is in very close proximity to the next enzyme, and it just enters that enzyme. And these massive, or I should say at least large enzyme complexes, are known as metabolomes, which are formed in conglomerates. And when glucose enters, it's not just being randomly found and then everything just randomly floats, but they all in a close proximity and more effectively pass the substrate one to each other. So I thought that was an interesting thing, a noteworthy thing to realize, because sometimes when we talk about this pathway, especially schematically, we just imagine these things floating around. They actually don't. They're in close proximity, and they're in heavy communication, close communication with each other, in close contact, to allow effective processing of metabolites. All right, enough about the citric acid cycle at this point. Now, this is going to be my last slide. I want to focus now on the glyoxylate cycle. This is one of my favorite cycles, and I don't know why. I just like it. And probably because we as mammals and as humans, we don't have it. And someone would say, well, why should we care if we don't have it? Well, it's not that we should, it's not that it's not important for us. It teaches an important principle, but more importantly, it helps us understand another principle that I hope to demonstrate by going through this. And that's why I enjoy talking about the glyoxylate cycle. So, what is the glyoxylate cycle? It is a pathway to convert fatty acids into carbohydrates, which doesn't happen in the human body, in human cells, or in any mammal cells for that matter. This happens usually in plants, invertebrates, and some microorganisms. Not in humans. What is it? You have acetyl-CoA that is fused by oxaloacetate, same reaction as TCA cycle, citrate synthase, we form citrate, then isocitrate, but from this point on, there are some differences. Now, in these microorganisms and in plants and invertebrates, there is an enzyme known as isocitrate lyase that converts isocitrate to glyoxylate and succinate. Glyoxylate then, with the help of malate synthase, converts another molecule of acetyl-CoA and glyoxylate to malate, which is converted by malate dehydrogenase to oxaloacetate, the same way as the citric acid cycle. Alright, so that's the glyoxylate cycle. Alright, so how is the fate of acetyl-CoA different in the glyoxylate cycle compared to the TCA cycle? Think about it. Now, in the glyoxylate cycle, we are fusing acetyl-CoA with the intent to make glucose. How does that happen? Well, here, we have acetyl-CoA is fused, another acetyl-CoA is fused. What's one of the products? Succinyl-CoA. Are there any other products? Well, NADH. So what would happen with the succinyl-CoA? Or with the succinate, I'm sorry. Well, here it shows that succinate actually then can enter mitochondria and be converted to fumarate, malate, oxaloacetate, and in the cytosol can be converted to glucose or fructose or whatever, and then from there to sucrose. That is the purpose of the glyoxylate cycle. To use acetyl-CoA, fuse it together. Look, for example, this succinate basically looks like two acetyl-CoAs fused together. That's what it looks like, two acetates fused together. But then that is being converted, or gets converted to glucose and sucrose. Alright, when acetyl-CoA goes through the TCA cycle, what did we learn so far from the TCA cycle? What happens with acetyl-CoA when it enters TCA cycle? The purpose is to burn it. The purpose is to break it down and make carbon dioxide and electrons. The purpose is completely different, completely the opposite. Alright, so the TCA cycle also produces succinate. Remember, that's part of the TCA cycle. Then why then the TCA cycle doesn't facilitate the conversion of acetyl-CoA into glucose? And this is, I haven't told you that yet, but you cannot make glucose from acetyl-CoA in mammalian cells. You cannot. even though acetyl-CoA can be converted to succinate, and we can see from here, obviously, that succinate can be converted, and normally it is, even in the human body, to oxaloacetate. To oxaloacetate and glucose. Then why doesn't the citric acid cycle do that? Why doesn't it convert it? Well, let's think about this. Entering of the citric acid cycle, entering of acetyl-CoA into the TCA cycle doesn't result in a net increase of oxaloacetate, which then can be converted to PEP and glucose. Instead, 2 carbons are released as carbon dioxide. And that is the reason why there is no net increase of oxaloacetate. Simply put, the glyoxylate pathway circumvents the first half of the TCA cycle, where the carbons are released. And because of that, it allows for actual conversion of acetate groups into oxaloacetate, PEP, and glucose. The glyoxylate cycle fuses, like I said, acetyl-CoA into succinate. It leads to an increased amount of succinate, and then oxaloacetate and glucose. The TCA cycle breaks acetyl-CoA into carbon dioxide, and it doesn't lead to an increase in succinate or oxaloacetate. So that's why the TCA cycle cannot be used for producing glucose. It is a cycle for burning. It is an oxidation cycle. No net increase. If anything, we just regenerate oxaloacetate. The acetyl-CoA that enters, in essence, is being burned to carbon dioxide and water. So this is the interesting part that I wanted you to realize, and this is the reason why I like talking about the glyoxylate cycle, because it demonstrates this sometimes difference between theory and practice. We're making succinate, we're making glucose, so yeah, theoretically speaking, we can do the same thing with the TCA cycle. Not really. We can't, unless we somehow bypass the first half of the citric acid cycle and invest into metabolites after the carbon dioxide release. If that happens, then we can. But if it doesn't happen, we cannot. And since acetyl-CoA enters at the beginning of the TCA cycle, we cannot use it to make carbon dioxide. We cannot use it to make glucose. Instead, we make carbon dioxide. All right. I hope this was interesting, and I hope this was clear and you enjoyed learning about it. That's all I have prepared for today. If you have any questions, please let me know. I'll be happy to answer them. Again, thank you for your time.